Hey guys, sorry I had to stop in the middle of the recording because I forgot I had to talk to my family members. So the second part video was kind of short. So there's just a few slides. Let's finish it for this chapter. I think we stopped here. So the termination of translation. So once the ribosome reaches the stop codon, remember the stop codon? 3 stop codons, right? And the UAA is one of them. Once it reaches the stop codon, that's a signal for the ribosome to stop translation. So the ribosome will disassociate from the mRNA. So that's the end of translation. So after the polypeptide chains have been synthesized at the end of translation, it's actually not the final protein product. Okay? It's very similar to mRNA. Remember, you have pre-mRNA. That's the end product for transcription, right? But that's not the final mature functional mRNA. So eventually they have to go through some modifications to become the mature mRNA. Alright, remember there are 3 major modifications: 5' cap and 3' A tail, and the splicing of introns. Okay, so similar proteins or the polypeptide chains will also have to go through some modifications to become the final functional protein molecules. So during and after translation, individual amino acids within amino acid chain may be chemically modified. So signal sequence may be appended and the new protein folds into a distinct 3-dimensional structure. So remember when we talk about the other 4 biomolecules, protein is one of them, right? And then we went through the different levels of structure. So from primary structure, that's the basic amino acid sequence. And the secondary, the alpha helix, the beta sheets, to tertiary, right? The interaction within one polypeptide chain, and then the quaternary. So if a protein has more than one polypeptide chains, the different polypeptide chains will kind of interact with each other, and that will form the entire kind of 3-dimensional structure of the protein. So the structure is so critical to protein function. If the structure is modified, then, or if it's not normal, so modified and now it's not normal, then the protein will become non-functional or dysfunctional. So it may become a very bad protein, like a disease protein, as in the case of Alzheimer's or mad cow disease. Okay, so what is a signal sequence? We mentioned this, I think, really briefly when we looked at the human insulin gene sequence. I remember the part that's in orange, those orange letters actually code for a signal. So the signal sequence is a kind of short segment of amino acids, and the job for the signal sequence is to kind of guide the protein to a specific cellular compartment to be processed. So once that's achieved, the signal sequence will come off the protein. So it's actually not part of the final functional protein. Many proteins can fold spontaneously, which is good. They don't need much help. But some other proteins require helper molecules called chaperones to kind of help them fold, to prevent the protein molecules from aggregating during the process of folding. So what does it mean? Remember, the protein structure is a 3-dimensional structure, right? So there's a lot of kind of interactions between different functional groups, a lot of folding involved. So if the protein can fold correctly, then that's good. But that's a very complex process, folding. So some of the polypeptide chain, the ends can be very sticky. So the molecules may tend to aggregate, and that's a very, really bad thing. You don't want them to aggregate to kind of clump. So this is where we need the help of chaperones. You have seen this term before. So the chaperones will prevent proteins from clumping so that they can all assume their correct structures. Okay. And this has application for us too, right? We don't want people to aggregate or to congregate now because of the pandemic. So make sure you engage in social distancing and then just stay at home. So just hang in there for a little bit longer. Okay. Right, so this slide is kind of continues on the chemical modifications and protein activities. So chemical modification can affect protein activity. So it can affect whether the protein is active or not. So these are the common chemical modifications. So a lot of times we can add functional groups to a protein molecule to change its activity. So examples of functional groups we can add it are methyl, remember ethyl CH3, acetyl, so now you have 2 carbons, okay. And then phosphate, right, PO4 with the 3 minus charge. The last one is ubiquitin group. So what is ubiquitin group? Now you see 2 protein molecules right here. So this protein has no ubiquitin tag. So this is almost like a tag. So this protein will remain intact and not destroyed in the cell. But once the ubiquitin group is added to the protein, now it's a signal for destruction. So the cell will know, okay, this protein is no longer needed and we need to degrade. We need to destroy this protein. So that's what a ubiquitin tag is for. The protein, once it has the ubiquitin tag, will be degraded by proteasome. So it's a cellular structure that contains the hydrolytic enzymes that can break down protein molecules. And this part, I'll let you read. It's pretty straightforward, and the information is very general, so just make sure you're familiar with it. Chemical modifications can occur in response to external stimuli. So it's not just internal signals, but also what the external environment is like. So stress, like nutrients, heat. The next one, epigenetic accessibility, we will talk about that in gene expression chapter, the next chapter. Okay, so you can just read that real quick. The last topic for this chapter is mutations. I really like this feature. I don't know if you guys like X-Men, but I like the movies a lot. So what is a mutation? A change in the nucleotide sequence. Remember, if you have CGA, let's say this is the normal sequence, and for some reason, there are a lot of factors that can cause a mutation. If the G is changed to C, then that's a mutation. There's a very interesting information here. So the mutation could happen to DNA. Everybody knows that. Or it may also happen to RNA, because a lot of viruses, their genetic material is RNA. And we know that viruses can mutate pretty fast, right? So that mutation actually takes place in RNA. So it can happen to both DNA and RNA. Mutation can be passed on from one cell or organism to another. So if one cell has the mutation, so this is the parent cell and it has a mutation. When the cell divides and becomes 2 new cells, because of 2 new daughter cells, let's say this is mitosis, they are the clone, almost the clone, right? Identical to the parent cell. So the mutation will be retained in the 2 new daughter cells. Okay? And then similar for one organism, the parent passing the mutation to the offspring. Implications for somatic cells and germ cells. So if mutation happens to somatic cell, you can have some diseases, for example, leukemia. If the mutation happens in germ cell, like sperm or egg, then the offspring will get mutation, and the disease is genetically inherited, like sickle cell anemia or albinism. So that's when the organism doesn't have too much pigment, so it looks very pale, like a albino squirrel, albino tiger. We can also have albinism in humans too. Mutations and protein functions. So mutation happens to DNA or RNA sequence, and we use those sequences to produce proteins, right? So you can imagine that if a mutation takes place, then it's probably going to have some consequences on protein functions. So there are all the consequences. First of all, you may have a silent mutation. So in silent mutation, the mutation has no effect, no impact on the protein product. So let's see over here. Oh, I have some examples later. So let me just go through the text and then we'll look at the examples. Another possibility is that you are going to have a loss of a function. Okay, so this mutation will cause a gene not to be expressed. Or it may produce a protein that no longer plays its role. So it's a non-functional protein. It doesn't do its active job. Okay. Alright, and then let's see the next one, gain of function. So this type of mutation will lead to a protein with a new function. Okay, so that's why it's a gain of function. Blah blah blah. Yeah, so we'll have some specific examples later. Point mutation. So this is a specific type of mutation. Definitions: addition and subtraction with a single nucleotide or the substitution of one nucleotide base for another. So there are two types, transition or transversion. So in transition, the original nucleotide, if it's a pyrimidine, then it's replaced by the other pyrimidine. So that's transition. And same thing for purines. Right? If you have A, which is purine, and it's replaced by G, is mutated to G, then that's also a transition. Transversion is going to be the change between pyrimidine or purine, or other ways from a purine to pyrimidine. So that's the two types of point mutation. Okay, so these are examples for the second part, right? The substitution. Now you may add or remove a single nucleotide. That's also considered a point mutation. I'll have some examples later. So we're going to focus on these 4 types of point mutations. Pause for a second. Okay, I'm back. This is a very important figure because it shows you the different kinds of mutations. I think it's really good and demonstrating the different types of mutations. So the one on the top, that's the normal sequence. And you remember we're using the genetic codons to determine what amino acid we're going to add to the polypeptide chain, right? Okay, so the only thing is that they're showing you the DNA sequence instead of the mRNA sequence. But you know that mRNA is pretty much the same as the DNA, right? So it doesn't really matter. Okay. And then we're going to do the codon tables and we figure out. So ATG, remember AUG is the codon for methionine, right? The start codon. But again, remember this is the sequence for DNA, not the sequence for mRNA. So this is why it's not AUG. Okay. And then you can see these are the amino acids that the DNA instructs to make. So first step, silent mutation. So in silent mutation, remember here, it does not affect the protein function. And why is that? Because a lot of times we have multiple codons that code for the same amino acid. So if you have a point mutation here, A is replaced by G, then even though you have this mutation, the codons, these two codons, the new codon also codes for leucine. So the amino acid actually doesn't change even though you have a nucleotide, one nucleotide replacement. So this protein has the same amino sequence as the normal one. So they're exactly the same amino acid chain. Okay, so there's no functional change. So that's a silent mutation. The next one is missense mutation. So in missense mutation, the mutation will cause will lead to a different amino acid. And because of the amino acid sequence is different now, the protein will probably have a different structure and different function. So that's why we call it missense. Okay, so let's look at here. So the mutation happens to here, right? So instead of a TTA, you have a TCA. And after you convert that to the mRNA sequence, the new amino acid coded by that codon is going to be serine instead of a leucine. So this is a completely different amino acid than the normal, amino acid protein than the original protein. Okay. So this is probably going to have changes in function. Alright, now nonsense mutation. So nonsense mutation is very easy to remember. Basically, the mutation causes or results in the occurrence of a stop codon. So if you change TTA to TGA, and then again after you convert that to the mRNA sequence of codons, this calls for the stop codon. So the ribosome is going to stop translation right here. So instead of amino acid chain that has 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids, now you have 5 amino acids. So that's nonsense mutation. Frame shift. So frame shift just means one base pair or one nucleotide is deleted. So again over here, so instead of a TTAC, now you have TAC. So this second T is deleted from the sequence. So now you're going to shift, because you're going to read 3 nucleotides each time, right? So you're going to completely shift how you read a codon, right? So now everything is shifted to the left. Okay, so that's a frame shift mutation. And again, because of this frame shift, you are going to make a different polypeptide chain. On the test, I'll have examples like this picture, something very, very similar to this, and then you have to identify what kind of mutation it is. Is it silent, missense, nonsense, or frame shift? Okay. Chromosome mutations, we're going to skip this part, so you need to study for that. And those are just some examples. You can look at them. This just kind of points out that not all mutations are bad. Of course, some mutations are bad. They can lead to cancer and lead to some genetic disorders. But some mutations are still good because they introduce genetic variation, right? Genetic diversity into the population. And this is also the basis for the species diversity, right? Through evolution, all those mutations accumulate, and those lead to new characteristics, and the population, eventually, a new species evolves. Okay? So just know that not all mutations are bad. I think that's the end of this chapter.